Hello everyone and welcome to today's webinar. My name is Chloe and on behalf of Oxford Global, I would like to thank you all for joining us today. Today's webinar is brought to you by Daya Bojic, Project Manager at Sartorius BIA Separations. After the presentation, we will lead into a live Q&A and you can submit your questions using the Q&A feature. The full recording of this webinar will be made available to rewatch on our secured website, and the details will be sent to you shortly after the webinar. Without further ado, let's begin.
Okay, hi everyone. My name is Daria, and thank you for joining this webinar. So this webinar will be focused on chromatography solutions for efficient and comprehensive characterization of EV preparation. So basically, I entered this field about 8 years ago. And from my perspective, at that time, we were all struggling with methods, and addressing even the most elementary questions seemed rather tremendously complicated and laborious. Since then, the methodology improved a lot. But I guess you will agree that some general facts about the EVs and EV studies still hold. So some basic features of EV samples include that they are normally heterogeneous, complex. There is never enough of the samples for all the experiments that are wanted to be made, and the samples are very variable. So the questions are always open. What are all the particles in the samples? Which subfraction of this heterogeneous population is relevant? How to isolate it? How well need the samples to be characterized? How many methods we should apply to actually get a good picture about what is there in our samples? So how to do this? How to reasonably select the methods to have enough sample for the basic characterization and the actual bioactivity studies. And what markers we can use to actually tell us the most essential information. So the current solutions of EV fields include the raising the awareness about the heterogeneity of the samples. So if about 20 years ago we used expressions such as exosomes, microparticles, ectosomes, and so on, this terminology was replaced by a broader term of extracellular vesicles to cover all the heterogeneous, like all the different origins of the vesicles in the samples. However, it was soon realized that even this might not be enough to describe all the particles in the sample. So the expressions such as EV enriched preparations was used instead. And nowadays we often observe that the terms such as extracellular nanoparticles is used instead of instead of defining which sort of particles we are dealing with. To address the complexity of the preparations, the field compiled the guidelines, which basically suggest to analyze the samples with as many methods as possible. This often means a lot of works, high costs, time-consuming analysis, and all this together often limits the study scope. Nevertheless, there is rarely enough of the samples to perform everything that we want to perform. So nowadays, we have many more sensitive and specific analytical techniques. Even single particle analysis is possible. However, all this sensitivity has some drawbacks. So we often lack representativity. And again, dealing with many different samples with various sources, we need the methods that can be applied on several different samples. There are also strong initiatives to standardize the procedures, to make the conclusions comparable among the studies. However, standardization is beneficial for the good methods. If we have poor methods, the results will remain poor no matter how detailed we standardize the procedure. So basically, the current methods, the established protocols, usually gives us a very tight insight into the samples. It is a little bit like playing Guess Who. We pose the question and get feedback from the analysis. So we need to ask, are they particles? Yes, there are particles. They are approximately this and this size. Are there proteins? Yes, there are proteins. Are there nucleic acids? Yes, there are nucleic acids. Are there vesicles? We apply electron microscopy and we can see nice vesicles. But even having all this information, we often lack a linkage between them, telling us what is actual relation between all these parameters we assessed. So to efficiently guide our process development, we seek a reliable and feasible approach, which will provide us as many information as possible in a reasonable time with reasonable amount of work. So the perfect method would be universal, allowing for analysis of a broad range of samples, possible to apply to the unknown samples. So it would need to have a wide dynamic range to give as many different perspectives to the sample as possible, be specific, but at the same time tell us a general integrated picture about the sample. So the aim of this webinar is to propose or to describe our approach using the chromatography as one such tool that can address some of these questions. So in the first part, I will describe our method, what information we can gather from analytical size exclusion chromatography. I will provide you with some basic general framework of where this method can be applicable, in what ranges it would provide reliable results. I will address some critical factors that need to be considered to assure reproducible and reliable analysis. And in the second part of the presentation, I will introduce you to two-dimensional coupled method that provides us a detailed insight into the extracellular nanoparticles heterogeneity. So, and at the end, there will be place for discussion, of course. So the basic principle of the characterization by size exclusion chromatography is to introduce the sample to the column that separates the components according to their size. The elution is then recorded by several different detectors we usually apply monitoring of absorbance at 260 and 280 nanometers to provide us some basic information about biological components such as proteins and nucleic acids. We have fluorescence detection for sensitive evaluation of fluorescent components and also evaluation of specific markers. And last but not least, multi-angle light scattering detector can be very efficient for sensitive particle detection and also allows sizing. So if we have a look to general sample fingerprint, the chromatogram would be compiled from different signals: absorbance, fluorescence, and light scattering. So just visual evaluation of such chromatogram already tells us a lot about the sample. In this case, I will show you a few chromatograms of different samples from purification process. So at the beginning, we have a crude sample, conditioned medium. We can see quite a lot of proteins being there. There are also some particles indicated by light scattering detector, but the amount of marker is really very low. So here we have the EV depleted sample. We can see some small components, including proteins, present, and while there are almost no particle detectable, at the end we have an example of EV enriched sample in which we can observe that proteins are largely removed, and while we have large amount of particles, so we can really already visually observe the enrichment of this fraction. So I would like now to go through the approach analysis step by step. So if we first have a look on absorbance, here we have two chromatograms showing separation of some protein mix for orientation about the size ranges throughout the elution. And the second chromatogram is showing an actual real EV sample. So at the beginning of the elution, we have particle population. Then we have macromolecules such as proteins, nucleic acids, and small complexes of them. And last components eluting are small components such as amino acids, vitamins, nucleotides, and so on. So the light absorbance for particles is usually relatively low. Also, the signal ratios would be affected by the scattering effects. But later in the elution, we can follow the proteins, and the assessment of integrated area below the signal actually allows us to evaluate free protein contents in the sample. Here I show you standard curve plot for an albumin. You can see that linear range for protein detection is really quite wide in this method. So if we need even more sensitive protein detection, we can go for intrinsic fluorescence of tryptophan. The next detector, I will describe the light scattering. So basically, light scattering detector is very sensitive for particles. Larger the particles, more intensively they scatter the light. Of course, also the refractive index is of importance here. So here we can see the NTA analysis of the sample that was injected on the column. If we have a closer look to the peak and compare it to the chromatograms obtained for the polystyrene size standard, we can see that some information about the particle size can be extracted already from the elution time. However, for more precise analysis, it's better to use multi-angular light scattering and actually assess the angular dependency of the signals. So if we have a look on the parameters obtained from the NTA analysis, we observe that actually the values obtained are relatively well comparable. So here we have the radius obtained from light scattering that relates very well to the NTA measurements. So light scattering intensity, of course, must be interpreted with caution, respecting the size distribution, as it's well known that the intensity is really strongly dependent on particle size. However, if we have the same population of the particles, light scattering intensity can be used to estimate the sample concentration. And the dependency of intensity is linearly related to the sample concentration. One of the very important questions in EV analysis is marker profiling. So one of the advantages of the size exclusion chromatography is that the bound probe is always already separated from the excess probe during the analysis. So this very efficiently minimizes the risk of increasing background due to the excess probe. However, the same applies as in other immunoassays. So the critical controls would include checking the autofluorescence of the sample and the stability of the labeling. Compiling the results of absorbance, fluorescence, and light scattering can already provide us some information about the heterogeneity of the sample. However, interpreting this discrepancy must be made with attention to the different physical background of detection for different signals. So if we now try to understand how the parameters that can be extracted from SEC analysis compare to other orthogonal methods. So here we have some results obtained by NTA, by SEC light scattering, with breadth photoanalysis for protein contents, integrated area from size exclusion chromatography for absorbance at 280, and the marker analysis. All these apply to the integrated area below the signals. So here we can see that the particle size estimations are very well comparable. Also, if we compare the enrichment factors for the particles obtained from the concentration obtained with NTA to the intensity of the light scattering at 90 degree angle, quite well aligned. The protein estimation from SEC analysis shows the same trends as analysis obtained from Bradford. And here we also see the evaluation of enrichment for different EV markers was quite comparable for the three probes. So from this perspective, we would come to the same conclusion about the efficiency of the purification process if we use several different methods such as NTA, Bradford, and a separate marker analysis such as use like ELISA, for example, or if we extract all the information from size exclusion chromatography. So I would like to address now a little bit the dynamic ranges. It is normal that dynamic ranges would vary greatly depending on the focus of analysis. Individual detectors have their own dynamic ranges, and of course, it will always depend on the sample and the parameter we want to assess. However, based on our experience, the usual EV samples normally fall within the range of the described detectors. So for protein assessment, based on albumin, we can estimate that limit of detection would be about 50 nanograms per milliliter based on tryptophan fluorescence, up to 1 milligram exploiting UV detector. Light scattering, of course, greatly depends on the particle size. However, a rough estimation of limit of detection for EV samples would be about 5e⁸ particles per milliliter. The fluorescence detector has the dynamic range with over 2 logs. So, of course, it would depend on the fluorescent intensity of the probe and amount of the marker present in the sample. However, here you can see an example in which we show that usually the fluorescence shows linear dependency within the range of approximately 2e⁸ up to over 1e⁻⁻. So basically, the linear range may be expanded even further, but who would want to use that much sample for analytics, right? So the next question would be how robust this approach can be and how to assure the reproducibility of the analysis. So one thing is optimization of the seq mobile phase. The running buffer would affect the interactions within the sample components. It should minimize the interactions between the column solid phase and the particles in the sample. It should provide the best resolution possible, and at the same time, it must maintain the probe fluorescence. So in this example, you can see how concentration of the salt in the running buffer impacts the resolution of the method. It is expected that increasing salt concentration reduces the interaction within the sample and the chromatographic column solid phase. And increasing resolution with increasing salt is expected. The running buffer may also slightly change the retention time of components. In this particular case, we can observe that it also affects the fluorescence. When using fluorescent probes, it is really very important that this possible factor is also considered. Here we can see the pH dependency of the FITS fluorescence. So when using immunolabeling or other fluorescent probes, it's good to those that are least dependent. As the conditions within the method are normally well controlled, the sensitive probes might cause the variation in the results between the different sequences and sets of analysis in case of variations in buffer preparation, or let's say ambiental temperature if we are using temperature sensitive buffers. So in setting up immunoassays, one thing is always addressed, and this is the probe concentration. Titration experiments are regularly performed with an aim to reach the saturation level and minimize the background in the method. A factor that is less frequently addressed is the time to binding at equilibrium. So to actually reach the stable binding, while this might not be that important in the assays such as ELISA and so on, when samples are where the readout is performed at the same time, this very strongly applies here where the different samples are analyzed sequentially. So one thing that I would like to also address is the impact of the sample medium on the binding efficiency and the sample stability, as also on probe fluorescence. So one of the critical things is really understanding the labeling kinetics. In these graphs, I show you the results for the same sample that was analyzed after different time points from spiking with the antibody. So here we can see that the absorbance and light scattering signals did not change within the experimental time frame. So the sample was stable. However, in case of immunofluorescence, we can see great increase of the signals in the first stage, in the first hours of immunolabeling. So actually, the equilibrium is established only after 15 or 20 hours. So actually, the time to equilibrium depends on probe concentration, the accessibility of the binding sites, and the affinity of the antibody. So with high affinity antibodies, the dissociation constant is small, and therefore the required time to equilibrium over 10 hours is not that unexpected. So the next thing I will address the titration, the probe concentration. So here we can see results of impact of the probe concentration on the signal intensity for 3 different EV markers. So the aim of titration is usually to reach the saturation level. So in this case, after reaching saturation, the further increase in probe concentration would not cause the further increase of the signal intensity. And we can be sure that we have optimal detection of the particles. However, it often occurs that with increasing concentration, the signal further increase even with very high probe excess. So in such cases, the further increase in probe concentration would increase the sensitivity. However, it would also increase the cost and ecological footprint of the analysis. Our experiments show, however, that relative signal intensity in relation to sample concentration gets linear at much lower concentration, so that reaching the saturation level of binding is not necessary to achieve linear response. However, the higher probe concentration may decrease the time to reach equilibrium, and in some cases increase the sensitivity. But as said, it is not crucially required to obtain linear response. So I would next like to address the sensitivity of the method to detect changes in the sample and sensitivity to the factors of the background media of the sample. So in this slide, you can see the characterization results for a sample spiked into different buffers, having different salt concentration and different pH. So basically, the sample was spiked in this buffer together with the probe and analyzed after a sufficient time. We can see that in relation to salt concentration, we have changes in immunolabeling intensity. We can also observe the increase in light scattering intensity and decrease in particle size. So if we would have an isolated information about marker presence and size of particles, we could assume that actually particle degrades at higher salt concentration. However, preservation of tryptophan fluorescence and even increase in light scattering intensity and preservation in one of the markers suggest different story. So in this particular case, we can link the decrease in size to the shrinkage of particles in response to hypertonic conditions rather than to the degradation of the particles as such. Here is another example showing intrinsically fluorescent engineered EGFP expressing vesicles. So here we can see, similarly as in previous case, the difference in the intensity of the tryptophan fluorescence in response to higher salt, decrease in particle size, while in response to the variation in pH, we can see that higher fluorescence is present at higher pH. So again, having an isolated information about the EGFP fluorescence, we might conclude that the particles are unstable in acidic conditions, while taking together all the informations provided from the chromatogram together with tryptophan fluorescence and light scattering intensity and size of particles, we can link this lower fluorescence to the sensitivity of the EGFP itself, rather than to degradation of the particles at lower pH. So I hope I convinced you that multiparametric approach to characterization is really beneficial in avoiding misinterpretations in our data analysis. So to really perform the reliable analytics and have all the suitable controls, we suggest that in any assay we include the system suitability test controls. This would normally be stable material, such as protein standard mix. We suggest using the reference sample to test the time dependencies, sample stability, and signal reproducibility. And then, depending on the focus of analytics, one should use all the necessary controls that are usually applied in immunoassay. And if one wants to perform sizing using multi-angle light scatter detector, a size standard should be included as well. So basically, such multi-parametric assessment then gives us a general view, a general picture about the composition of our sample. And such analytics may be applicable in the sense of assessing concentration, recovery, purity, contaminant profile, marker profile, and stability of the sample. So I would like now to quickly also describe our further more advanced techniques to actually even further analyze the heterogeneity in the particle population. So the first step of this method would be the same as described, but in this process, we capture the particles and direct them to another column in which they are separated according to their charge. So the result of such two-dimensional analysis would be a complete profile of components resolved by their size and the heterogeneity of particles resolved by their charge. So here we can see an example of the EV sample of engineered CD63 GFP EVs. We can see the profile of the impurities in the sample and the particles resolved by their charge. We can notice several populations, like we can notice two main populations of EGFP expressing particles, which could be EVs. And we additionally observe the tryptophan peaks, which suggest that there are also some protein-rich particles present. So if we spike this sample with the antibodies, we can follow the shift in peaks in response to the antibody probe binding. This is expected as the binding of the probe would change the surface of the particle. And this can actually allow us to assess the possible overlapping populations in the sample. The peaks that remain unaffected by the probe suggest the presence of the particles that do not have the targeted marker. So in this slide, you can see a comparison of a few EV samples. Already visually, we can see that the first samples are much less pure. And the further assessment of the particles can tell us that they have different populations of particles and that some similar populations of marker positive populations are present. Whilst the marker profile of particular subtypes of particles is different in each sample. So the main advantages of such two-dimensional approach is that a general and very detailed profile is obtained in less than 1 hour. It requires minimal sample preparation, as the sample can be injected directly to the SEC. SEC would remove all the possible interferences for the further ion exchange examination, and would also provide us, and the further ion exchange examination would provide us the information about the heterogeneity of the particles in the sample. For such approach, we can use about 100 to 300 microliters of the sample, depending on concentration, of course, but this would usually allow for analysis for obtaining the basic profile, even for the crude clarified harvest sample. The approach is applicable to very broad range of samples, and it allows us to identify different subpopulations of EVs and non-EV nanoparticles that would naturally overlap in size. It would also allow us to quantify and profile non-particle associated macromolecule residual, and we would also already get some information for potential further purification stages. So, to sum up, we find this approach very convenient in assessing production of EVs, purification processes, optimizing the formulation, assessing stability of the sample, and the same approach can be applied also for optimization of loading and functionalization. So basically, this would be my last slide, and I would like to now to take any questions if there might be. So thank you.
Wonderful. Well, thank you. Thank you very much for your presentation. As you just mentioned, we will now open the floor for discussion. If you have a question, please submit it using the Q&A feature, and Dea will be able to answer it for you. So we already have a few questions. So this came in during the start of the webinar, so you may have already touched on this, but somebody has asked for some more information related to plant-derived exosomes. If you're able to share any information.
For plant-derived samples, yeah, actually we have in past analyzed some of those. The plant-derived vesicles are kind of particular because there is not many specific probes available, but we already obtained some profiles using some membrane dyes. So yeah, it can be applied. Is there any additional, more specific question on this particular topic?
The same person has just asked, can we load drugs into plant-derived exosomes? If you wouldn't mind answering that one.
Yes, certainly I don't see the problem. I guess, I think there are also already quite some publications available on this topic, but in our group, I think we haven't obtained such samples for analysis, so I hardly tell more about this.
Thank you for your answer. We have another question. So, what are the biggest current bottlenecks you see in EV isolation and characterization workflows?
Yeah, actually, as I said, usually we have a very, very particular, we are so focused on trying to find EVs in the samples that we kind of forget about everything else that is present in the samples. So basically, we are lacking a real and firm linkage between the components of the samples and their roles, their contributions to the bioactivity, their functions. I see this as the main drawback, yeah, and the complicated analytics.
Fantastic, thank you very much. Another question, so is there a risk of EV aggregation or loss during the SEC analysis, and how can this be minimized?
Yeah, well, we performed some assessments of the recovery of the particles during our SEC method. And I can tell you that for our past samples, we normally observe the recovery of about 85%, which is, in my view, rather good. Like, of course, there is some risk of damage of the particles. One can address this by lowering the flow rate of the analysis, decreasing the shear stress on the particles, really optimizing the running buffer conditions, and yeah, maybe like I would go for these aspects as first. But in our experience, this is not such a very big issue, the stability of or damage of the particles within the analytics.
Okay, thank you very much for that answer. So another question, how adaptable is the SEC method to different EV sources such as plasma derived versus cell culture derived vesicles? Apologies for my pronunciation.
So I can say that our current approach works very well for cell culture samples. Of course, blood plasma is more complicated, and one needs to consider also all the components in it. So we don't have that much experience with plasma samples, but for sure the running buffer should be optimized for this particular, like with addition of some anticoagulants or protease inhibitors. I don't know, I'm just guessing. And the solid phase of the chromatography column might be selected according to the properties of this particular sample. Yeah, hope this answers.
Thank you. Another question, so how can you distinguish between EV that shrunk versus degraded? And does shrinking reflect an issue with potency or functionality?
Yeah, basically, what we find important is taking into consideration the different information from different detectors. So if we observe the decrease in size, we would expect the decrease in light scattering intensity as well, if the decrease in size is the consequence of degradation. In this particular case, we observe increase in light scattering intensity, as well as increase in tryptophan fluorescence. So this is contradictory to the hypothesis that the decrease in size is linked to the degradation. Also, if we have a look to the fluorescence in this particular case, we can see that the fluorescence did not change the fluorescence of the particles. So it appears that all the particles that were there are still there. So really connecting different parameters is crucial to actually draw better grounded conclusions. And yeah, about if shrinkage affects the bioactivity, I simply don't know. This is something that needs to be assessed. And also, one possible control of whether it goes for shrinkage of the particles or actual degradation is also, yeah, one can dialyze the sample back and observe if the particles swallow, and one obtains again the same size as before shrinkage or not. So yeah.
Thank you very much. So another question. So is it better to lyophilize the exosomes after isolation or use it as such? And at what pH of PBS are they stable?
Particles after lyophilization. Huh. This is a hard one, actually. Well, I have not that much, I don't have very much experience about the stability during lyophilization processes. This is, I guess, one thing that could be assessed by this methodology. And my guess would be that the stability of the EVs in respect to the pH would depend on their source. So it's hard to say, to define a range without better knowledge about what samples we are talking about. Yeah.
Thank you. So we have a few minutes left, so if you want to ask any questions, please feel free to ask them now. We do have another question. So is it possible to use the new SEC methacrylate column offered by Sartorius BIA separations to analyze the EVs?
Yes, indeed, it is possible. One needs to actually, it's important that one selects the best column for the purpose. The columns offered by Sartorius are good ones and well can apply to various samples, but it would like really depend on the samples. Yeah, methacrylate columns are applicable to wide range of samples, so I don't see the reason why not. Yeah.
Wonderful, thank you. And then this may be our last question. So the chromograms display signals for various markers. Is it possible to incorporate multi-labeling into the described approach?
Yeah, basically, like our setup allows detection of at least two different fluorescences. So, and if needed, another detector again with another two fluorescence channels can be also added. So basically, the methodology allows multi-labeling. What might be limitation is the number of available binding sites. If you are talking about the immunolabeling, I would like the same issues appear as with other methods such as flow cytometry, for example. So in introducing several different probes might complicate the interpretation of the results, but I don't see such problem in case using, let's say, one surface protein marker targeted by antibody and another probe maybe passing the membrane, so membrane permeable to assess the particle carbo. So yeah, but
Thank you very
otherwise,
much.
yeah,
Oh,
for
sorry,
assessing,
continue.
yeah, sorry, for assessing surface markers, I would suggest to assess them one by one before going into immunolabeling, for sure.
Wonderful. Well, thank you very much for your answers. We're now at time, so unfortunately that is the webinar. I want to thank everybody for joining and Daya as well for their presentation. If we didn't get around to your question, the team will follow up with you shortly after the webinar. And as mentioned at the beginning, the on-demand recording will be made available over the coming days, so please do look out for this in your inbox. And thank you again for joining. Have a lovely day.